next organic substance, lipids. Lipids are insoluble in water. Here are some examples: triglycerides, phospholipids, and steroids. Lipids' main function is to be used for cellular energy, so that's for long-term storage of cellular energy. And that's long-term. Lipids have more energy per gram than carbohydrates. Carbohydrates have about 4.5 calories per gram, and lipids have about 9 calories per gram. So lipids have twice as much energy. The carbon, like carbohydrates, lipids are mainly made of carbon, hydrogen, and oxygen, but there's a lot less oxygen than in carbohydrates. And generally, lipids have a molecule of glycerol and 3 fatty acids. So the monomers, or the building blocks for lipids, are glycerol and fatty acids. There's an example of lipids. You've got saturated fatty acids, and saturated fatty acids are like the ones from animals. These are not great for us, and well, we do need them, but they're saturated with hydrogen ions. So that's what it means to be saturated. Every spot that can have a hydrogen ion has a hydrogen ion. Unsaturated fatty acids, like olive oil, be a good one, are not saturated, so they don't have a hydrogen ion every place. And because of that, they take on this little bit of a curvature, this shape, and that's what allows unsaturated fatty acids, without getting too deeply into this, to be more healthy, because basically they can't be stacked, quote unquote, in the same way. Okay, and then there's the glycerol portion of a lipid molecule, and then there's the fatty acid portions. Okay, phospholipids. Phospholipids, you just ignore these two pictures. So we can ignore those for right now. Don't worry about these so much. Just look at this one. Phospholipids have a hydrophobic tail. They have hydrophobic tails and hydrophilic heads. And because of that, phospholipids are really good for forming our cell membranes. Okay, so hydrophobic tails and hydrophilic heads, because the heads are attracted to the inside of the cell and the outside of the cell. We'll see that more in the next chapter. Steroids do important things in our body as far as hormones and helping to make up our cell membrane, but we need steroids as for our sex hormones, so testosterone, estrogen, progesterone. Okay, on to the third organic molecule, proteins. Proteins are used to make structures like our muscles, also enzymes. They're also an energy source for us. Actually, I'm going to stop here and then make proteins. Nope, we'll keep going. So let's go on with proteins. So proteins used as an energy source. They're building blocks. Their monomers are amino acids. I'm going to abbreviate amino acids as capital A, lowercase a. And amino acids each have an amino group, which is an NH2 group, a carboxyl group, COOH group, and then whatever unique side chain that makes each amino acid different. There's 26 amino acids in nature, and we use 20 of them. Humans use 20 of them. So you can see that there's the general formula, but here's an example. So cysteine has its COOH group and its NH2 group. Okay, and then this right here is the R side chain, and that's what makes cysteine different from all the other ones. Same thing for phenylalanine. See how phenylalanine's R group looks very different than the cysteine? Okay, this just points out that proteins are bonded, or amino acids are bonded together by things called peptide bonds. So peptide bonds will hold amino acids together so you can form those longer polypeptide chains in those proteins. There's 4 levels of protein structure. They're over here. Primary level, this means primary. The primary level is just the sequence of amino acids. Okay, so it's each amino acid in a row. Secondary level is either a beta pleated sheet or an alpha helix. Okay, a beta pleated sheet looks like this, and the alpha helix looks like this. Tertiary structure is how the protein is folded. Okay, and then each tertiary structure for protein is different, and it's really those more distant interactions that help to fold it that way. The tertiary structure is so important for enzymes because enzymes have to be folded a certain way so that they can have an active site to work on their substrate. So in the tertiary structure is very sensitive to differences in pH or temperature. So if it's in the wrong pH, the tertiary structure can unfold and make the enzyme useless. Quaternary structure is just the number of proteins or number of polypeptides in the protein. Okay. So primary is just the sequence of amino acids. So amino acid 1, then amino acid 2, bonded together with those peptide bonds, so on and so forth. Okay, well, finally we've got nucleic acids. Nucleic acids are things like DNA and RNA. DNA functions in carrying our genetic information and in protein synthesis. And RNA just functions in that protein synthesis for the most part. So DNA carries genetic information and protein synthesis, and then RNA is just protein synthesis. Okay. Building blocks of DNA are nucleotides, and this is a nucleotide which has a phosphate group, some base, one of 4 bases. So there's 4 bases. They're adenine, guanine, cytosine, and thymine. You don't really have to know the names right now, but those are the 4 bases. And then a sugar. For DNA, the sugar is deoxyribose. It's a 5-carbon sugar. For RNA, it's ribose. So DNA has the sugar deoxyribose. DNA has the sugar deoxyribose. So it's a 5-carbon sugar that's missing an oxygen. And then RNA has ribose. Okay, this shows that DNA is a double-stranded molecule. So you've got nucleotides on both sides that pair up And we're not going to really get into what the pairings are, but each of these is complementary to the other. If you remember probably from high school biology, if you've taken biology in college, adenine always matches up with thymine, and cytosine matches up with guanine. RNA is single-stranded. So some differences in there. RNA's single-stranded, has ribose as its sugar. DNA is double-stranded, and it has deoxyribose as its sugar. Okay, and this just gives you more information. It kind of just goes over what we just talked about. Okay, so take a look at that. Make sure you have all that information down. Science to technology, CT scanning, and PET imaging. So CT scanning is computerized tomography, or CT imaging, and it uses X-rays in a computer to make a three-dimensional image. Okay, and this would be how we would detect a tumor, would be a good use of a CT scanning. Positron emission tomography, or the PET scan, uses radioactive isotopes in a computer, and those isotopes, we would add isotopes into the person, and when those isotopes go someplace and they're metabolized or they're broken down or they're being used or incorporated into tissue, the radioactivity will give off a signal that's picked up by the computer, and you can see, for example, a PET scan is useful for seeing what part of the brain is active during some activity. So maybe you want to see how the reward center in the brain is stimulated by a certain activity. You would use the PET scan, and when that reward center, when you're watching the person doing the activity, if the reward center lights up, it means that center's using energy, and that would show up on the PET scan.